All right, first part of this is going to be review. Toward the end, we're going to get to some stuff that we haven't gotten to yet. You got to know the equation for photosynthesis. If you don't know that, this unit's not going to do very well on. We know plants need light. What's the word for like the smallest piece of light that can exist? A photon, not a proton. Photon. It's just a piece of light. It's the smallest piece of light you can get. So plants need photons. They need light. Obviously, they need some carbon dioxide. How does that get into the plant? How does the CO2 get into the plant? Stomata or stomates. Stomata. And those stomata or stomates are located mostly where? Bottom of the leaf, the underside of the leaf. They also need water, which they take in, obviously, through their roots. They use those two things to make sugar, carbohydrates, which we may write as like glucose, or we may shorten it up to that, CH2O, sugar, carbohydrate. And then in addition, we know they make some oxygen gas. Do plants need oxygen? Yes, because they like you, have to do what? Cell respiration. They do both. And remember, it's not just plants that can do this. Other than plants, what other things are photoautotrophs? Other than plants? Lena? What group is that? Algae, and also plants, algae, and some of the bacteria, the cyanobacteria, can all be photoautotrophs, do photosynthesis. So know the equation. That's number one. Know the reactants for photosynthesis, and know the products of photosynthesis. Now, mostly what we talked about so far was the very first part of photosynthesis, the light-dependent part. And remember that the light-dependent part happens in the chloroplast. Specifically, it happens on these little pancake-like shaped discs. Those pancake-shaped discs are called the thylakoids. Each disc is a thylakoid. Waiting on you two guys to pay attention. A whole stack of these thylakoids is called a granum. A whole stack of thylakoids is a granum. That's where the light-dependent part happens. The light-independent part, the Calvin cycle, happens out here in the space, fluid-filled space. And that fluid-filled space is called the stroma. Now, this is the way it works in plants and algae, because they're eukaryotic. They have chloroplast. In bacteria, they don't have these, right? Where does photosynthesis happen in a bacteria? Where are all the proteins at in the bacteria? The proteins involved in photosynthesis? In the cell membrane. So the light-dependent part happens on the cell membrane. The light-independent part, what's the only thing left? Cytoplasm, just the middle. So it works a little bit differently in bacteria. And what we're going to talk about here first is what's called the light-dependent part, or non-cyclic photophosphorylation. I'm going to put in a blank slide so I can draw my picture up here. All right, now remember, what we're drawing here is a picture of a thylakoid, a single one of those little pancake-like structures. Here's our thylakoid. Embedded in that thylakoid are groups of light-absorbing pigments. And we call those groups of light-absorbing pigments, what's the name for them? Photosystems. And they're numbered backward. Photosystem, this is photosystem 2. My marker's a little off up at the top. And this is photosystem number 1 over here. In the middle of each photosystem is a molecule of chlorophyll 8. It's called the reaction center. We want to think of these photosystems, though, like big groups of what? What's a photosystem like? What human machine? Like a big group of solar panels. And all these solar panels are taking that energy that they absorb, and they're funneling it to these guys here in the middle, these reaction centers. Eventually, all that absorbed light causes these reaction centers to do what? What does all that energy that they absorb make them do? Shoot out some electrons. They become what's called photoexcited. But before we get to that, in between the photosystems are these guys called cytochromes. And they make up what kind of system? What do the cytochromes make up? Electron, what? Transport system. They carry electrons. So the electrons get lost. Here comes the electrons. Both photosystems lose electrons. Photosystem 2's electrons hop across the cytochromes. And as it's moving, what are those? The energy from those electrons is used to power what? The pumping of H+. And H+ in this case gets pumped from the stroma out here into the middle of the thylakoid. H+ getting pumped in. And we're building up a big gradient.
From the stroma?
From the stroma into the middle of the thylakoid. What's the word for the middle of any tube? The lumen. Okay, so you get pumped into the lumen of the thylakoid, the middle. And these things want to get out, but they can't. Because what's the membrane made out of? Phospholipids, and they're ions. The ions and phospholipids don't like each other much. They're stuck. The more we pump in, the more they want to get out. The bigger the gradient, the more energy we're storing. All right, photosystem 1's electron, the one that it lost. It combines out in the stroma with this electron carrier called NADP+, and also some hydrogens to make what? NADPH. To make NADPH, we had to do what to NADP+? Reduce it, which means we added energy to it. This is a form of stored energy. Energy that used to be where? Sunlight. This is like bottled sun energy. Now, these H+, they need to get out. So that's where this comes in. This is, what is that? ATP synthase. It's a enzyme and a what? Channel protein. It's a channel for what? H+. When it opens, these H+ shoot out. The enzyme part of that channel uses that energy to take ADP plus P plus that energy to make what? ATP. And that way of making ATP, what's it called? Chemiosmosis. Using energy from the H+ gradient to make ATP is chemiosmosis. So far we've made two things, NADPH and ATP. Both forms of what? Energy that used to be sun energy. The last thing that we're going to make is this photosystem right here, number 2, needs it to get an electron back. It lost electrons, it needs to get them back. Where does it get them back from? Water. It basically mugs the water molecule, rips it apart. It steals the electrons from it. What's left behind from a mugged water molecule? Well, H+. If we did this twice to 2 water molecules, if we mugged 2, that'd leave 4 H+, right? And it would also leave 2 O's. But what do we know about O atoms? They get together, they partner up. So O2 gets made, and that O2 eventually gets out of the plant, most of it, through what? How does it get out? It's the stomata, the little holes on the bottom of the leaves. So during this process, the light-dependent reactions, 3 main things get made. Oxygen gas, ATP, and NADPH. This whole picture is sometimes referred to as non-what? Non-cyclic, because what's not moving in a circle? The electrons. They're basically starting at water, and they're going from water to photosystem 2, across the cytochromes, to photosystem 1, and ending up where? In NADPH. They're not moving in a circle. That's the non-cyclic part. What's the second word? Non-cyclic what? Photo, because of light, phosphorylation. All right, it's called that because the energy from the light causes ADP to get phosphorylated in order to make ATP. Non-cyclic photophosphorylation. That's that process. That's one way of doing the light-dependent part of photosynthesis. Photophosphorylation. Now there's also a cyclic version of that looks like this. Most things that do photosynthesis are doing both of these at the same time, cyclic and non-cyclic. Ricky, join us here, bud. The cyclic version only involves photosystem number 1. When light hits it, its electrons get ejected. They eventually move in a circle, but they come back to where they started. As they're moving, the electrons cause H plus to get pumped and ATP to get made. But since the electrons end up back where they started, what doesn't need to happen? What never gets made if the electrons end up back where they started from? NADPH, and there's no need to rip apart what? No need to rip apart water, so no oxygen gets made. So this non-cyclic version makes no O2, it makes no NADPH. And I got my letters wrong here, NADPH. It only makes ATP. The reason that's important, to make sugar, plants need a little bit more ATP than they need NADPH. They need both, but they need more ATP. This lets them get that extra ATP that they need to make sugar. Now, another question. That's the light-dependent part. Makes ATP and NADPH. Those are forms of energy. The definition of photosynthesis is taking light energy and converting it into chemical energy. We've done that by the end of this step, right? Why bother making sugar if you've already done it right here? What do we say about ATP and NADPH? That makes them not good for making food. They're both really reactive, which means a plant can't do what with them if they're really reactive? Can't store them for later, and also can't what? Can't move them around. There's almost all photosynthesis happening in the plant. The leaf. Well, the stem needs some food. The roots need some food. So the plant has to be able to take this ATP and NADPH and make it into something that it can ship. What's the thing that it can ship? Glucose or sucrose, sugars. So that's the purpose of the Calvin cycle, is to take the ATP and the NADPH and to make them into something that it can actually store and ship around for later. Now, we talked a little bit about the Calvin cycle right before the break. Let's start off by looking at this picture. Now, that looks kind of complicated, I know, but it's not so bad. The Calvin cycle is all about carbon dioxide. If you remember, carbon dioxide has very little energy. That's why we breathe it out after cell respiration. We've gotten all the energy out of it we can. It's kind of just a waste. But the plant breathes in that carbon dioxide. Plants, all plants, have an enzyme called rubisco, sometimes also called RUBP carboxylase. This word RUBP carboxylase, we'll see why it's called that here in just a second. What? It should be in there. Yeah, it should be in there. So the plants take the CO2, and this enzyme, rubisco, the most common enzyme on Earth, grabs onto that carbon dioxide and attaches it to this 5-carbon thing called ribulose biphosphate, RUBP. So carbon fixation. That just means grab the carbon and attach it to an organic molecule. So rubisco grabs the carbon dioxide and attaches it to this thing, 5-carbon RUBP. That's why it's called RUBP carboxylase, because it adds a carbon to RUBP. Now here's the thing. Notice this is happening 3 at a time. 3 CO2s, 3 RUBPs. We take 1 carbon plus 5 carbons. That gives us this thing, a 5-carbon substance. But it's really reactive, and it breaks up. So we have 3 6-carbon things that break up into 6 3-carbon things. Don't make the math harder than it is there. We got 3 6-carbon things. Those break in half into 6 3-carbon things. These we're going to call PGAs. It's like half a sugar, but half a sugar without what? What's missing from this thing? What are we putting in here and here? Putting in some energy. Okay, so this is like half a sugar without the energy. It's like diet sugar, right? Sugar without the energy. So right here we phosphorylate PGA. Phosphorylate means add phosphate, which brings with it energy. So we're adding energy. Where did this ATP come from in the first place? Where did the ATP come from? What did we just talk about? The first part, light-dependent part, right? This was made in the light-dependent part. So now we're taking the energy from the light-dependent part, we're putting it into this sugar. All right, notice right here. What's happening to NADPH? Right there. NADPH to NAD plus is what? Oxidation. Which means what's happening to this thing? It's getting reduced. Okay? It's getting energy and electrons that used to be where? Sun energy. So during the Calvin cycle, we're taking the energy from light-dependent parts, putting it into a sugar. That's called the reduction phase. So in the Calvin cycle, phase 1 is grabbing the carbon, fixing it to an organic molecule, carbon fixation. Part 2 is taking the energy and the electrons from the light-dependent part and adding it to that CO2, reduction. By the time we finish, we end up with 6 3-carbon things called G3Ps. Those are like half of glucose. They're half of glucose with the energy. So that's like the high-calorie sugar stuff. Remember, we got 6 of those. We take 1 out. The other 5 keep on going. The other 5 are used to do the last part of the Calvin cycle, which is the regeneration of RUBP. These have to get turned back into this so the cycle can keep going. Seems kind of wasteful. Made 6, only use 1, but that's the way it works. When this goes through 2 times, 2 G3Ps together basically make what? A sugar, a glucose. So G3P and G3P make glucose, basically. That's the Calvin cycle. Now, a couple of things that are really important that you need to know about. Every plant does this. Every plant has this enzyme. That enzyme is the most important, most common enzyme on Earth. But that enzyme has some problems. Why it evolved this way, we don't really know. That enzyme only works really well when what is going on inside the plant. When there's a lot of what in the plant. What does the plant need to do? Calvin cycle. Carbon dioxide. Rubisco only works well when there's a lot of carbon dioxide. How does the plant get the carbon dioxide in the first place? How does it get in? Through the stomata. When it's taking CO2 in, what's coming out? And they're just little holes. So CO2 is coming in, what's coming out those same holes? Oxygen, but more importantly, to a lot of plants, what? What else could, if you had big holes in your skin, what else could come out? Water. That's a problem when it's hot and dry, right? It's losing a lot of water. So what do plants sometimes have to do with those stomates? Close them to keep in the water, keep them drying out, desiccating. Well, what I'm telling you is when they do that, this enzyme, it doesn't work very well. I'm going to make an analogy. We're going to make an analogy to Rubisco as a cheating husband. Rubisco, let's say he's married up to carbon dioxide. That's his wife. And as long as she's around, he only has eyes for carbon dioxide. But when she goes away, What else is around? As carbon dioxide is getting used up, what is there becoming more of inside the leaf? Oxygen. So as CO2 goes away, he starts looking that way. Oxygen. Okay, Rubisco starts to instead of fixing the carbon dioxide to this RUBP, he starts grabbing onto oxygens. And I didn't mean that analogy to go that way. But he starts grabbing onto it, not the way I meant to go with that. But he starts to be attracted to oxygen. And he starts to attach oxygen to this RUBP instead of carbon dioxide. Well, if you attach oxygen to this, is that ever going to lead to sugar? No. It's a problem. And it turns out that problem is called photorespiration. When most plants get hot and dry, they have to close their stomates. They start fixing oxygen instead of fixing carbon dioxide. And that means that's a waste. That doesn't make any sugar. That's called photorespiration, when they fix oxygen instead of carbon dioxide. And what that means is they're just wasting energy. That's a very big problem for plants when it's hot and dry. That's the only time they're going to close up their stomates, right? Hot and dry. Most plants have that problem. Turns out most plants are called C3 plants. And the reason they're called that is because the first thing that they make during the Calvin cycle is this 3-carbon PGA that we've been talking about. That's why they're called C3s. There are some plants, though, that don't have this problem. And that's what we're going to get to here in just one second. Before we get to that, though, let's look at this picture. Inside the leaf of most plants at least around here, C3 plants, it looks like this. On top of most leaves, what is there on the outermost part of the leaf? Think about the outermost part of the leaf. What does it look like? The top of the leaf. Before the green. What's on top of the green? What's on the top of most leaves, guys? Think. It's kind of waxy. It's got a waxy coat on it. It's called the cuticle. That cuticle, that waxy coat on the leaf, is sort of like what on you? It's like your skin. Its job is to keep what in the leaf? Moisture in and what out? What does your skin keep out? Germs, bacteria, viruses. Plants get sick too. So the cuticle is to keep moisture in and germs out. Just underneath the cuticle, you've got all these cells. These are your photosynthetic cells, the cells that do photosynthesis. This whole middle part of the leaf is called the mesophyll. What's meso mean? Middle. So that's the middle part of the leaf, the mesophyll. The actual photosynthetic cells are called parenchyma cells. Those are the cells that do photosynthesis. They're called parenchyma cells. Now I want you to notice something about these cells. Notice up here at the top, they're really tightly packed together. But down at the bottom, they're all spaced out. There's all these holes in there. Why does it need to be that way? Why can they be tight at the top and loose at the bottom? What's got to come from down here and go all the way up to there? CO2, because what's that right there? And that right there? Those are stomates. Those are the breathing holes. If it was tight down here, the air could never make it up to the top, right? So these are like little passageways for the air to go through. CO2. It's got to be like that. Now also notice that running through the leaf are these things. They call them, on here they call them veins. Do plants have blood? No. But what kind of vessels do they have? What are the two kinds of vessels in most plants? Xylem and phloem. Those are inside these veins. One of the kinds of tubes, the xylem tubes, carry what? Water and minerals, mostly from where? From the ground up toward the leaves. What's the other kind of tube? Phloem. Cell and pH. And the phloem is carrying mostly what? Sugars. And it's mostly going in which direction? From leaves to roots, right? going the other way. They're going opposite directions for the most part. This is what most plants look like inside the leaf, C3 plants. And remember, these have a problem when it gets wet. When it gets hot and dry, they start doing what? Well, they close their stomates first, and then they start fixing oxygen instead of CO2. That's called photorespiration. And that's because that enzyme, rubisco, is like the cheating husband. Parenchyma, those are the cells that actually do the photosynthesis, the photosynthesizing cells. All right, but it turns out there's another kind of plant. And that other kind of plant is called C4. It's called C4 because the first thing it makes during its Calvin cycle is a 4-carbon substance. The first thing it makes after carbonization is a 4-carbon substance. Notice it's inside of its leaf looks a lot different. It's still got a cuticle out here, just like the other plants. But notice that the photosynthesizing cells make these big, long, big circular structures. They basically make a big, thick tube in here. Here's the deal. These C4 plants have 2 different enzymes. Out here in the mesophyll, they have, not in rubisco, they have an enzyme called PEP carboxylase. And we're going to say that this enzyme, PEP carboxylase, is like the always faithful husband. Only has eyes for who? CO2. It fixes CO2. It fixes CO2 with no attraction for what? No attraction for oxygen, which means there's not any problems with what? Photorespiration. Plant adultery, right? There's no problems with that. No problems with that. Which means these guys can do what and still be okay? They can close their stomates. This is still fixing carbon, no problems. It works even when CO2 concentrations are low. But here's the thing. It fixes the carbon out here in the mesophyll. Then that carbon that it fixes gets moved into this tube in here, this bundle sheet. It's like a little bitty tube inside a tube. The carbon gets turned loose in here again. And guess what enzyme's inside this little tube? That's where rubisco is at. Since it's in a little tube, even a little carbon makes what happened to the concentration inside that little tube. It makes it get hot. So what we're saying is that these C4 plants fix the carbon in one place and do what in another place? What happens in here in the bundle sheet? They fix the carbon out in the middle of the leaf, the mesophyll. They do the Calvin cycle inside that little tube that's called the bundle sheet. So they fix the carbon in one place, they do the Calvin cycle in another place, and that lets them not have such a problem with what? With drying out, with photorespiration, desiccation. Let's go to another slide and get a little bit more detail on this. Most plants around here are C3 plants. These kind of guys, C3. What are legumes? You've probably heard that word before. What's a legume?
Isn't it like a it's like a seed or something?
It's like a bean. So these are like bean plants, soybeans, peanuts, stuff like that. Notice all these things though. Those are the main what? Food crops, right? Main food crops for humans are C3 plants. And they all have a problem with what? Drying out. So most food in the world gets grown in places that's not too hot or dry. And remember, the disadvantages of C3 plants is when they start to dehydrate, what do they do with their stomates? They close them. What does that do to photosynthesis in those plants? It pretty much stops it because they start fixing what instead of what? Oxygen instead of CO2, photorespiration. They can't do much photosynthesis then. It just doesn't work right. C4 plants have an adaptation. They're evolved to deal with hot and dry. They've got that enzyme PEP carboxylase out in the middle of their leaf, the mesophyll. It fixes carbon even when what's going on. Even when stomates are closed and CO2 is what? Low. That's an advantage. Once they fix the carbon, they ship it into that tube-like structure called the bundle sheath. Turn it loose. What's inside the bundle sheath? What enzyme? Yep. Rubisco, RUBP carboxylase. And from there on out, The Calvin cycle works here just like it worked over here. Same thing. Some examples of C4 plants are these guys. Sugarcane, corn, Bermuda, sorghum. Around here, when is corn ready to pick in Alabama? When do you pick corn around here? July and August mostly. What do you know about July and August in Alabama? It's the hottest, driest part of the year, right? Corn can get by with that because it's a C4. Most people's yards around here in August, what do they look like? Most people's yards. Brownish. Because what kind of grass do most people have around here? Most people have grass around here called fescue. Fescue is a C3 plant, which means when it gets hot and dry, they start dying out. But some of you have this kind of grass in your yard, Bermuda. And even in August in Alabama, what does that Bermuda grass look like? It's green. It's still growing fast, and you're having to cut your grass like crazy. That's Bermuda grass. It's a little bit more expensive than fescue. It doesn't grow as well when it gets cooler. This fescue grows better when it's cooler. This grows better when it's hotter. What about sorghum? What's that? Anybody know what sorghum is?
It's kind of like wheat, isn't it?
Kind of like wheat. What do we use it for here in Alabama, in the South, mostly for? Speak up there, Walker. Molasses. You guys ever had molasses? It's kind of like, looks like black honey, sort of. It's actually really good, but it's from grain. But the thing is, what you need to know is these C4 plants have an advantage. They fix carbon in one place. They do the Calvin cycle in another. Their carbon fixation and Calvin cycle are said to be spatially separated. That lets them be able to live where? Hot and dry. Now there's one other group of plants that's even better. They're called chem. They fix the carbon only at night. Their stomates are closed all day. These are things that live mostly in deserts. What's it like in the desert during the day? Really hot, really dry. What's it like at night in the desert? Kind of cold. Okay? So their stomates are closed all day. They're wide open all night. During the night, they're using PEP carboxylase to fix the carbon at night. They store it in vacuoles at night as an acid. It's stuff called malate. During the day, they close those stomates. They turn that CO2 loose. But enzyme fixes it during the day when the stomates are closed. Rubisco. So they have the PEP. That works at night, storing carbon dioxide as acids. During the day, there's a lot of CO2 in the leaf because it just keeps getting released. All that stuff they fix at night gets released. And Rubisco does it from there. These guys are the best at living in hot, dry places. They've got two adaptations. They've got the better enzyme, PEP carboxylase, for working that when the stomates are closed. And they only fix the carbon when? At night, which means they don't have to worry about losing water. These are things like cactus. Also lilies and orchids and pineapples. Where do pineapples grow? I mean, what kind of plant? I always thought until the last 10 years or so that they grew on trees. It just seemed to make sense. Okay? Now, one day, years ago, I was watching Survivor TV show, and they walked into this looked like some grass, and they said, "Hey, here's some pineapples." And I thought, that's crazy. Hey, pineapples grow on trees. But they were growing little shrubs about yay tall. Not tall at all. And turns out that's where they grow. They grow little shrub-like plants on the ground. But they're chem plants. They're said to do the carbon cycle, or the carbon fixation and the Calvin cycle are said to be temporally isolated from each other. What's the word temporal mean? What's temporal mean, Jeff?
Short time.
It means time. They're separated by time. They fix carbon at night, the Calvin cycle during the day. That lets them close their stomates during the day That lets them not worry about losing water during the day. So these plants are the best in terms of dealing with hot, dry. Here, this is showing you C3. Remember, C3s are the ones that have a problem with photorespiration. They take in the carbon dioxide, they use rubisco to attach that carbon to RUBP and eventually make G3P, what we call G3P, sugar. They run into problems when it's hot and dry, and they start having photorespiration problems. Now, on the other hand, C4 plants fix the carbon out in the mesophyll with PEP carboxylate. And remember, it works even when what's low. Even when CO2 concentrations are low. So these guys can close their stomata, keep working. Once they fix the carbon, they pump it into this enclosed space called the bundle sheath. Turn it loose. Inside the bundle sheath is rubisco. And from there on, it works just like C3 plants. They fix the carbon in one place, do the Calvin cycle in another place. And then the CAM plants, they temporally isolate carbon fixation in the Calvin cycle. They fix the carbon at night with PEP carboxylates. Store it in vacuoles as an acid called malate. During the day, that carbon is re-released into the plant as carbon dioxide. Their stomates are closed though. But there's plenty of carbon dioxide because this stuff they stored during the night is giving it off. Here they use rubisco and they do the Calvin cycle just like everybody else does, just like all the other plants. So again, in CAM plants, carbon fixation and the Calvin cycle are said to be temporally separated. Separated by time, when they do it.
Does CAM stand for something?
Yeah, it stands for Crystalline Acid Metabolism. You don't even really need to know that. Just know CAM plants. Know that CAM plants are like cactuses, for the most part. All right, let me get this recording taken. Go clip.